Talk me through this whole Sirtuin thing. That's a pretty broad question.
Yeah.
Let me narrow it down a little bit. Talk me through what you were just about to allude to. What was Lenny onto when you showed up?
Well, genetics is a fabulous tool because you don't have to go in with any hypothesis. That biology will tell you the answer. And the gene that had just been cloned by Brian turned out to be what's called SIR4. Now, SIR4 didn't turn out to be a mammalian conserved gene. So SIR4 has not been as exciting, but there was this partner of the SIR4 gene. It was called SIR2. And SIR2 and SIR3 and SIR4 form this complex of proteins, stick together and control, of all things, gene silencing. That's what SIR stands for, Silent Information Regulator, in this case, number 2. And that was very unexpected. In those days, if you think back, the cause of aging was thought to be DNA damage, mutations, free radicals. So we were all expecting to find genes that controlled DNA repair or antioxidants.
At that time, because I go back and I think, God, at that time I was still in college studying math and engineering. I hadn't taken a biology course, so I can't think about it through the context of my own education. How well was it understood that there were introns and extrons and that much of the genome wasn't even coding and stuff? Like, was that well understood at that point in time?
It was. In yeast, actually, they were ahead of anybody else in eukaryotes. We knew that there were introns. There are not many introns in yeast anyway. But we hadn't seen the full genome. It was still bits and pieces. Maybe 5-10% was known. My PhD was sequencing 3 genes. That's all it took. Things were changing rapidly. This new thing with PCR, you could move tubes into hot tubs and amplify genes. It was all very exciting. But what we didn't have any clue was that why a silencing gene that negatively controlled genes, other genes, why that would have anything to do with aging. It was totally bizarre. It led to a string of cell papers. And one after another, every few months, we were actually publishing something new, and we had a science paper. And that was the gold rush of this discovery, because it really turned on the lights in this cave of a whole new area of biology. And we're still trying to understand actually why those silencing proteins are relevant to aging. But what we do know for sure is that these same genes, these Sirtuins, in all life forms, whether they're from plants all the way to us, do play a protective role in responding to energy and nutrients, just like the AMP kinase and the mTOR pathway do.
So you alluded to Sirt2. Was that first found in yeast?
Yes, it was. It was known already, actually. Lenny, and we didn't discover Sirt2, it was already known as a silencing protein that controlled the mating type. In other words, the sex of a yeast cell. And if you don't have this silencing, what happens is that the yeast cells get confused because they're now turning on genes for A and alpha, which means male and female. And a yeast cell that doesn't know if it's male or female will not mate, and it's become sterile. And it turns out that's a hallmark of yeast aging, is sterility. So if the way you can tell whether a yeast cell is truly old is it sterile or is it just sick? And that's how we used to tell. But now we actually understand the cause of that sterility. It's the movement of Sir2 protein away from those genes that it should be at to go deal with other problems in the cell.
I see. So it's not that Sir2 becomes deactivated. It just, for lack of a better description, shifts its attention elsewhere?
Right. It becomes distracted by other things going on in the cell. And we had a cell paper in 1999 with Kevin Mills and Lenny where we discovered, and a couple of other groups should also get credit for co-discovering this, is that the Sirtuins are also involved in DNA repair. When you get a broken chromosome, it's the Sir2 complex that goes along, helps unwrap the DNA, we think, and put it back together and repair that. And while the Sir2 complex is doing that, it cannot be also silencing. There's not enough of it to go around. And you might ask, well, why would the cell do that? Why don't you just make more Sir2? What we think is that this is a very ancient system that coordinates controlling mating and DNA repair. You don't want to be mating and dividing if you've got a broken chromosome. So this is a way of coordinating those two events. And it's very ancient. It's a very active system. You need DNA checkpoint signaling, so it's not just random. But what we also have come to realize is that this, let's call it this distraction of the Sirtuins, it's conserved. We find this happens in our own aging process as well.
So there's really two roles. There's gene silencing and DNA repair. Now, Sirtuins are HDACs, is that correct? Are they all HDACs?
So HDAC, histone deacetylases, this is the old name for protein deacetylases now. Because what we've all come to realize is Histones are just one of the things that sirtuins and these other HDACs do. They can target what are called non-histone proteins, and they remove not just acetyl groups, but also other types of what are called generally acyl groups. And so that's a whole new world. That means that sirtuins, the family, there are 7 of them in mammals, 5 of them in yeast, target other proteins that are in the cytoplasm, in the nucleus, even in the mitochondria. And that's their role. It's not so much only about controlling the chromatin and histones, but also about controlling signaling and metabolism as well. And they can do that by targeting any protein theoretically in the cell.
So what are the, again, thinking back to DAF2, DAF16 as the parallels with FOXO and IGF, were there elegant experiments in the yeast that could show you extreme conditions of lots of SIRT, no SIRT, and what that phenotype is?
Oh, yeah. These were the first experiments. So I'll try to take you through them correctly in sequence. So what we showed with Brian, first of all, and this is a 1996 Cell paper, was that we were looking for this movement. There was this so-called age locus. We didn't know what it was. We didn't know where they were going. We just knew that they left the silent mating type locus. So what we did was we stained it. So Brian had moved on actually to his postdoc, and Kevin Mills and I, student at the time, our job was to find where are the SIRT proteins going. So we stained them and we could look at them under the microscope. And what we saw was they were going to this little place in the nucleus, which we eventually figured out was the nucleolus, which is what makes the rRNA, which makes the ribosomes. It's a really important part. And the DNA that's within the nucleolus is called the ribosomal DNA or the rDNA. And that's where they were going, not just during normal aging, but also during an accelerated form of aging. So there's a whole story that was lost in history, actually, that maybe I'll just quickly touch on. One of the first things I did when I got to Lenny's lab was to work on Werner's syndrome, which is premature aging disease.
And these are kids that die in their teens or 20s, aren't they?
No, that's a different Hutchinson-Gilford syndrome. This one, Werner's, they live till 40.
They live in their 40s, yeah.
But the gene was just cloned by George Martin and his team out there. And the homologue in yeast is called SGS1. And I picked up the paper, it was in Science, I recall, went into Lenny's office and I said, I've just been scooped, but there's a yeast homologue. I'm going to work on that. Is that okay? And he said, yeah, go for it. And so we worked on SGS1 for a little bit. And what we found was that they were going through accelerated aging as well. So we had a science paper on that. And what was exciting about that was that they were also becoming sterile and the SIRT2 complex was moving as well, just like the normal aging. So we had this model, rapid aging model. And you might say, well, so what's the big deal? You've got a rapid aging model. But that told us that there was some universal process that results in premature aging in humans, probably in yeast, the same thing. And we could also study this much more easily than an old yeast cell. Consider that to find a single old yeast cell, it's really hard, especially if you're just studying replicative aging, which is the number of times they divide. Each mother cell produces, on average, 10 to the power of 25 offspring.
Wow.
Okay, so that's, by my calculations, about 30 million offspring. And you have to pull out that one cell and study it biochemically.
Wait, you said 10 to the 25.
Yeah.
That's
2 to the 25.
Oh, okay.
Yeah. But in any case, that's way more. So what we used to do is to sort out the old cells. We'd label them with a chemical and pull them out with magnetic beads. But it was we couldn't get many of them. You'd just get a handful. But with these SGS Werner's proteins,
you could get a bunch of them.
The mutants, we could get a lot more. And so we'd made a lot of progress using that. But every time we made a discovery with the SGS protein mutants, we went back to the normal yeast cells and verified. But we were actually able to figure out with that mutant what the distracting problem was for the SIR complex. And that was actually DNA breaks and DNA recombination that was occurring at the most repetitive regions, the most unstable region of the genome, which is the rDNA, which is in the nucleolus. And that was what was distracting those proteins.
Interesting. When you knock out SIR2, is it easy to knock it out?
Yeah, so that was the next experiment.
Okay.
So then we wanted to know, the prediction is if you knock out SIR2, you should get a lot more of this instability at the rDNA.
Yes. And the question is, does it translate to accelerated aging or not necessarily accelerated aging, but more cancer or some other phenotype, right?
Well, in yeast, it led to accelerated aging through the process I was telling you about. Mm-hmm. Genomic instability, DNA repair went down. And also that happens in animals, although it's a little more complex because it's embryonic lethal in a lot of mice. So you can't easily do that experiment. But what you can do is the opposite. you can turn on or overexpress the SIR2 gene in a yeast cell. And if we're right, you should get a few things that are going to happen. You'll have more genomic stability at this, particularly at this rDNA locus in the nucleolus, and the yeast cells should live longer. And that experiment was done by an incoming graduate student, Matt Kaberlein. And what a fantastic project to get when you walk in. He did it, and the day that he got lifespan extension with extra SIR2 was a very good one for him and the lab.
So what's the next step from there? The most obvious thing is, how do you develop a compound that would do this without the genetic mutation that empowered it?
Yeah, that was the issue, because you can't easily genetically manipulate humans. So the question was, how do you turn on these genes? Now, we spent about 3-4 years working on caloric restriction in yeast and then in mammals. And my lab and some others were leading the charge in showing that sirtuins, both in yeast and mammals, were not only necessary, but were sufficient when you overexpress them to mimic calorie restriction. Put another way, if you knock out SIR2, you don't get the benefits of calorie restriction.
You don't get the benefits of CR.
Right. And that's also now been shown by others to be true in mice as well. So that led to the idea that...
Is that true critical. In all mice?
I couldn't say. There's even in yeast, you can get around the need for sirtuins if you stress the yeast really intensely with very little amount of calories. But there are aspects of calorie restriction benefits, such as a lifespan extension, that are ameliorated, lessened by a SIR2 knockout, but it's still complexified by the fact that it's lethal in embryos, and you have to knock it out in the adult to do a real experiment.
Yeah, it's the same sort of issue that Cynthia had with the DAFs, which was if you do too early, the C. elegans doesn't make it into an intermediate stage.
You know what? Somebody hasn't even done the proper experiment, which is to take a mouse that you can knock out SIRT1 in an adult, whole body, and then calorie restrict. Something that we probably should have done years ago. We didn't. But the technology is there to do that. But what we did learn actually was, both in yeast and in mammals, was that it's not just one of these genes that's important. It's the whole family. And that if you knock out, take yeast for example, if you knock out SIR2 in yeast, you lose the ability to respond to some mild calorie restriction. But if you really calorie restrict them, they'll still live longer. And there was a big debate actually between Brian, Matt, myself about that. And where we settled on was that these other SIR2 related genes were also helping, and they work as a family. And if you knock one out, the others can compensate.
The yeast only have one?
Yeast have 5.
Yeast have 5. And humans have a whole family, like
7.
8 or something, right?
Just 7.
Yeah. Oh, okay. So only 2 more. That's interesting.
Little known fact, most people ignore the other yeast sirtuins, but they're just as interesting. And what we've found is they also can extend lifespan as well.
So when you... Actually, that's funny. I was going to go back to something else you said a second ago, but what's the teleologic explanation for why caloric restriction and sirtuins would move hand in hand? I'm just like, from you talked earlier about your appreciation for sort of evolutionary biology. So an organism is, in a nutrient deprived environment, it still has to be able to do a bunch of things if it's going to be fit. Do we believe that that's an environment where we need to see more stabilization of the genome, or repair, or silencing, or what do we think is the biggest insult during that period of time?
Yeah, so the biggest insult to any life form is a broken chromosome. That's lethal if you don't fix it.
And do we see that more likely happening during nutrient deprivation? I would see that as, I would have assumed, to be honest with you, I would have assumed that was independent of nutrient exposure, or at least, if anything, inversely correlated.
If you don't have enough nucleotides to complete replication, you're going to break a lot of times.
I see.
So they do go hand in hand. But the bigger picture is that the sirtuins evolved, we believe, what are we talking about, 3.5 billion years ago in the first life forms, early life forms. Maybe just after one of the first proteins to actually evolve, we think would be a sirtuin. And its job is to sense stress, biological stress in the environment, whether it's DNA damage, or it's a burst of cosmic rays, change in temperature, or a lack of nutrients. And their job is to allow that organism to hunker down and survive. Stop mating, stop breeding. We can do that in another day. If we don't survive this, our offspring are going to die anyway. So they control. We think they control which genes to turn on and off in response to adversity, and they allow those organisms to survive. But they're also talking to other pathways. So they're going to talk to mTOR, they're going to talk to Cynthia's DAF pathway. And collectively, these are the genes that we've settled on as the longevity pathways. But they didn't evolve for longevity, they evolved for survival during adversity.
How conserved are these across let's use the big four models of eukaryotes, from yeast, worms, flies, larger mammals like mice and rodents. Is this relatively well conserved the way the TOR pathway is conserved, or does it have more bends in the road?
Well, they're surprisingly conserved, that you can just manipulate one gene in each of these organisms and get lifespan extension, or one drug works in all of these organisms. I would challenge anyone to use a chemotherapy to help a yeast cell. So this is quite a magical discovery, that the same pathways are that well conserved and that ancient. And that's actually one of the advantages we have. Aging is actually not that difficult to be able to control, and that's because our models are very good. I truly believe that if we can extend the lifespan of a yeast, a worm, and a mouse, humans are so close.
Right. If you can do it across 1 billion years, you should be able to make that leap to a few other 100 million years.
Exactly. It's really just the regulatory agencies and making sure that we don't do any harm and it's safe. But the biology is all still there, going way back 3.5 billion years ago.
So it was 2006, 2007. When did resveratrol emerge as an early sirtuin activator?
2003.
Okay.
And the story behind that was that we were looking for an activator. We're hoping for an activator.
Now, were you in your own lab at this point?
Yeah.
You had finished your postdoc, I'm guessing?
Yeah, I managed to move to Harvard in 1999. So that was the year when a lot of things happened. While I was moving, we published this DNA repair connection.
But just going back to a silly sort of social question, did you at some point think, I want to go back to Australia and set up a lab here? Like, was it a difficult decision for you to stay in Boston as opposed to, because you came from Sydney, if I recall.
Right. Well, the goal was to,
because those are pretty different climates. I've been to Sydney once. I spent 2 weeks there. I could stay there.
Yeah, I miss that. But I also like adversity. I thrive on adversity. And so I've come to like living here. The intention was to come here for 2 years. I had a job to go back to. But the first week of being here in Boston was like a city I could only dream about. This is the Athens of ancient Greece, Rome of ancient Rome. For biology, this is it. And so I was in heaven. I'd never experienced a city where you're on the train and people around you are reading Science magazine and Nature. That's my dream. So it was a very easy decision not to go back to Australia, for reasons that if you really want to change the world, you got to do it from here.
So now you're in the process of kickstarting your own lab, which comes with its own stresses, right? You've got to secure funding and all those other things. And then what's happening on the front of this stuff? Yeah,
so 1999, a few major things happened. One was this DNA repair connection. The second one was Lenny's lab published that NAD was a requirement for sirtuin activity. And Shinemai, who's at WashU now, was the postdoc who made that somewhat serendipitous but brilliant discovery. And then the third thing was the connection to calorie restriction was happening around that time too.
Going back to the second thing, we talk about it now today like it's in textbooks. It's so obvious, right? That sirtuins are NAD-dependent deacetylase. Okay. NAD is so ubiquitous in cells that if the quantity you could have in a cell varies on a scale from 1 to 10, what is sufficient to produce this deacetylase activity? Is it anywhere from 2 to 10, or does it have to be, quite a high concentration?
It can be 2 to 10. You can actually get very low levels in some disease conditions, and the animal is still alive.
So it's basically only saying that NAD is necessary for sirtuins to work.
Right. Well, without NAD, we'd be dead in 30 seconds. But
by other reasons as well. I mean, exactly. Wouldn't we be dead just from not being able to do electron transports?
We would, and there's more than 500 reactions that we need just to survive. But what we didn't know in the 2000s, and it was actually quite a crazy thing to think that NAD was regulating anything. And that was actually what we first worked on in my lab, was the control of sirtuins with NAD levels. The reason it's crazy is you read textbooks, and NAD is the most ubiquitous, important molecule in the cell. How could it possibly be varied during aging, let alone during the day when you eat something or your circadian rhythm? Now it's obvious. We know NAD goes up and down. It changes with age. But in those days, people thought if you changed NAD levels, you'd probably die. And that's not true. Was it known at the time that doesn't complex 1 of the mitochondria basically convert NADH to NAD? Mm-hmm. So you would at least know that in the mitochondria, the concentration of NAD must go up and down, or else you couldn't actually respire. Well, locally it goes up and down, but the steady state level's pretty constant.
And when you say going up and down, are you talking cytoplasmic, nuclear, plasma? Where are we talking about?
Yeah, we had a paper, I think it was 2007, where we had quite a surprising result, which told us that it's not just the cytoplasm that it goes up and down. It was also the mitochondria going up and down. And we didn't know that. We stumbled upon it. We found that if we kept cells with high amounts of NAD, they would survive better, DNA damage, other insults. And we could deplete NAD in the cytoplasm, but the cells still lived. And we didn't understand that.
How did you do that, by the way? How do you deplete NAD?
Actually, come to think of it, we could either overexpress NAD depleting enzymes, but actually the way we found it was that when you damage cells with a DNA damaging agent, say chemotherapy, drug, the cells themselves deplete NAD naturally with this enzyme called PARP1, which is an NAD consuming enzyme. And that's very well known that if you hit a cell with a DNA damaging agent, the reason that it dies is NAD depletion. So we were measuring the NAD depletion in the context of...
And that's because the cell is trying to utilize that NAD to repair the damage you've just caused?
Right. So
or you didn't know that at the time?
No, that was well known. So PARP1 is a known DNA repair protein. And that's, if you block PARP, you also protect cells. But what was interesting was that we could overexpress, give more copies of a gene that made NAD. And this is called an MPT, which is the equivalent of the PNC1 gene in yeast, which we found was important for lifespan in those organisms. So we were overexpressing this MPT. Cells had more NAD. We hit them with the toxin. They'd survive better than the regular cells. But the NAD was still being almost completely depleted from the cytoplasm.
Sorry, just because this is sort of at the crux of it. Did they survive because they were unable to deplete their NAD?
No, we saw that NAD just disappeared from the cell, but they survived.
But they still survived.
But there was a place we weren't looking at the time. And we tracked it down to the mitochondria. So this begs the term, the mitochondrial oasis hypothesis. So myself and Anthony Sauvé from Cornell coined this term because what we found was that as long as the mitochondria stayed active with their NAD, it didn't matter. The cell could survive and recover from that stress. And actually it turns out that the levels in mitochondria, NAD levels, are even more important than cytoplasmic NAD levels for survival.
That makes sense. And of course, that's a hard thing to measure, isn't it?
It was extremely hard. That's where Anthony came in. Anthony is a chemist and a biochemist at heart. And it was extremely hard to isolate these mitochondria, preserve the NAD in them. We had to use new technologies to be able to do that.
Well, how was that done?
We were using mass spectrometry for the first time to measure NAD. And what Anthony did brilliantly was to make labeled versions of NAD and its precursors. And he could spike those in and use those as references to measure NAD levels in these compartments.
And NAD, if I recall from just biochemistry, you don't get to move that in and out of plasma into cells. It's made de novo in the cell. You have what you have. You can make more. You can bring in precursors, but you don't get to shuttle NAD between cells, correct?
As far as we know, right.
Okay. Yeah, it's sort of like an ATP problem. Like it's really hard to quantify ATP in a cell. By the way, does NADP do any of this as well?
Well, it's important, no doubt. It's part of this whole problem that we're working on. But if you add NADP or even NADH, which is only different by one hydrogen,
yes,
those don't work to activate sirtuins. Only NAD plus will do that.
And I mean, not to get too nerdy on this, but is it? It's obviously more than just the charge, but the charge must play a role if NADH can't work, right?
Yeah.
So it's probably exactly a combination of the charge and some other
and size. Yeah.
Size, yeah. Okay, so now you've made these three discoveries, the second of which we just went into in a little bit more detail. How does the story unfold now? You've got your spanking new lab.
Well, we worked for a little while on the SGS1 Werner's protein and telomeres, put out a paper on that. And we didn't work on sirtuins for about a year because Lenny said, I don't want you working on sirtuins when I left the lab, which was a bit of a shock.
Why?
He didn't want the competition, I suppose. But I thought a year was enough to give him a head start. But we quickly started working back on that.
Is that a common request of people when postdocs leave their labs?
No, of course not. But what am I going to do? I mean, the guy trained me.
Yeah.
I owe him my career. But in those days, it was very competitive. Mm-hmm. And so, the Lenny of today is not the Lenny of previous years, and he's a wonderful friend and mentor to me. But that's how it was in those days. It was very cutthroat.
I see. But if it were up to you, you would have continued to work on sirtuins.
Right.
So then now we're basically back to 2001, 2002. The moratorium is up. You're now back to working on it. And nobody at this point in time has yet figured out a way to exogenously manipulate sirtuins more than the stuff that we've already talked about, which is nutrients, stress, and things like that?
Yeah, we were trying to feed yeast NAD, and we gave them nicotinamide, which is a precursor to NAD, vitamin B3. And actually, Kevin Bitterman, who's now a very successful venture capitalist, my first student, he put nicotinamide on yeast, and we could measure the Sirtu activity by the color. And if Sirtu was more active, they'd turn red. And he walked into my office, one of his first experiments when we worked on Sirtuins, and he said, "David, something's weird. We didn't get activation, we got inhibition." So the yeast had gone red. And I said, "Kevin, doesn't matter what happens. If it's unexpected, that's even better." So that led to a paper that said that vitamin B3 high doses is inhibitory of sirtuins. And now the labs around the world use nicotinamide as an inhibitor for sirtuins. And I wouldn't recommend taking really high doses of nicotinamide.
Why is that?
There's a few answers. Biochemical answer is that there's an evolved pocket in the sirtuin structure that measures nicotinamide levels, and it's a feedback loop. So nicotinamide is, to get nerdy, is the product of the reaction. It takes NAD, cleaves it.
Oh, I got it. So it's just a negative feedback loop.
Negative feedback
it's
loop,
seeing too
right.
much B3, and it's saying, I have too much of my output.
Slow down.
Turn it down.
Exactly. So we struggled with that. We couldn't get NAD to go into cells. It's too big. Even with mammalian cells, it's difficult. So we were looking at ways to make more NAD in the cell. That was our original thesis before resveratrol was on the radar. And we were turning on and discovering the genes that made NAD in yeast. And we cloned some of the genes in that pathway. And there was one particular one that was called PNC1. And it had been studied in the context of tuberculosis. And what we found was that when we calorically restricted yeast cells, this was one of the most highly upregulated genes in the whole yeast cell, which was very unusual. People had discovered this before in their own lab, but they were wondering, what the heck is this NAD synthesis pathway? Got to do with calorie restriction, got to do with stress. But we knew exactly what was happening. This is a stress response that was turning on NAD production and activating sirtuins. So we had our first Nature paper actually on that, 2002, I think, 2003. And we found that PNC1 could mimic caloric restriction and raise NAD availability. And then if we knocked out the PNC1 gene, yeast cells didn't live longer when we calorie restricted them. And what's really interesting about that, I think, is that PNC1 doesn't just get turned on by caloric restriction. It's turned on by heat, low amino acids, salt, high salt. And so this is a gene that senses the environment and turns on the sirtuins. Exactly what I was explaining earlier about those early life forms on the planet, sense their environment, and through NAD and other ways, they can turn on these pathways for defense.